Okay, students, welcome to our lesson for Tuesday. Today, what we're learning about is carbohydrates. This lesson is taken from your textbook, page 29 to 32, and we are going to complete questions page 38, 1, 2, and 3. So let's start by breaking down what carbohydrates are. Carbohydrate contains two words, and oftentimes we don't break it down this way, but it helps us understand what's going on. Carbo stands for carbon, and obviously hydrate stands for water, or H2O. So when we talk about carbohydrates, what are we talking about? We're talking about simple sugars. We're talking about starches. These are the main things we would talk about in biology. It's also going to include cellulose as well. That is also a carbohydrate. And all carbohydrates contain carbon, hydrogen, and oxygen, and they're in the 1-to-1 ratio. Now this is cool because when we talk about carbohydrates, you can see the carbon and you can see the water right there. So there will be a certain amount of carbons. For each carbon, there's going to be one H2O that's attached to it. And because it contains this water molecule, it's going to be formed in the hydroxyl side group. You'll see a lot of hydroxyls attached to carbons. And this means that it's polar. And it also means that it's going to be easily dissolved in water. This is why your piece of paper here, this is a piece of cellulose. And when you put this in water, it absorbs a lot of water because it itself is made up of these polar bonds, and it's going to mix and interact with that water really well. So carbohydrates are the raw materials that we use to build amino acids, which are going to form proteins for us. Lipids, which are going to form our fats, and nucleic acids, which are going to form our genetic material. So where do these carbohydrates come from? Where does starch come from? Where does cellulose come from? Well, it comes from glucose, C6H12O6. Let me back up there again. C6H12O6. And where does this come from? Well, this comes from photosynthesis. Okay, so if we take CO2 and water, the carbon, we combine it with water, we do our photosynthesis. And you should remember this from grade 10 science, but CO2 plus water will make our glucose, and it will also make oxygen as a byproduct in photosynthesis. This happens in all green plants in the chloroplast. Now, once we get this C6H12O6, this glucose, we can polymerize it. So we can do these anabolic reactions. Remember, anabolic means to build. So we can take these glucoses and we can make starch, glycogen, cellulose from these monomers, these monosaccharides. Monosaccharide means simple sugar. So glucose is a simple sugar. And again, we're doing anabolic reactions where we're removing water. And it requires energy for us to do that. So let's talk about the structures and functions. There are three types of carbohydrates you need to know about. Monosaccharides, mono means one. Disaccharides, which means two, or oleosaccharides, which means more than two. And polysaccharides, which means many. So when you have one sugar like glucose, that is a monosaccharide. Ribose is another single sugar, and fructose is another one as well. When you take a glucose and you take a fructose and you combine them together, you get a disaccharide. There's two sugars attached to each other. Now, this one particularly happens to be sucrose, and that's table sugar. When you take more of those glucoses or fructose or riboses and you combine them together, you can combine them in different ways, and they can be used for several different things. One is your energy storage, so your glycogen in your liver. Another one is structural support. Plants use cellulose as their main structural support. So if you look at bark, that's cellulose. It's also used in cell-to-cell communication. So some of these things are used on the surface of cells to communicate between cells. Starch, which is an energy storage thing in plants. Glycogen is what's stored in our liver. Cellulose and chitin. So cellulose is used in plants. Chitin, we're going to learn, is used in shellfish. And it is made out of sugars, and it's just a slightly different bond. and you go from an energy storage source to a structural support. So here we have glucose, and this is linear glucose to the left here because it's in a straight line, and you'll notice it is an aldehyde, and the carbonyl group is at the end. And what happens is it's dry. It likes to have that linear form, but these double arrows mean it can switch back and forth. And what happens is it will actually form a cyclic ring here. And this has happened when glucose is wet. And this is the most common form of glucose that you'll see. Now, something interesting about glucose that occurs is that it will form two forms we call alpha glucose and beta glucose. And they almost look exactly the same. They're isomers of each other. And what an isomer is something with the same formula but a different structure. And these different structures completely different functions generally. Okay? So when you have different structures, you'll have different functions. So alpha glucose behaves differently than beta glucose. We're going to talk about some of those different impacts in a couple moments here. So let's look at the three-dimensional glucose to understand this further. So first of all, glucose again it looks like a hexagon. It's got an oxygen in that 6 ring, and we start numbering just to the right of that. So these are the carbons that I'm numbering right now. So there's 6 carbons in both of these molecules, in both alpha and beta glucose. If you look, you'll notice that the only difference in these whole structures is this little shape right here. And this is showing you that in this 6 ring, the 6 atom ring, the OH is below the ring, and in beta, it's above the ring. Let's look at it in 3 dimensions to see if we can see it clearer. Okay, this is a 3-dimensional model of glucose. Let's look at it closely. So here, if we move it around a little bit so you can see the dimensions, you can see that there is dark gray atoms that are represented by carbon. The light gray atoms are represented by hydrogen, and the red atoms are represented by oxygen. So if we look at the ring, we can see we have an oxygen here, and to the right is our first carbon. And if we look down, what we see is this oxygen that's pointing down, this OH. So the first OH is pointing down underneath the ring. The second OH is also pointing underneath the ring. So this is down, down. And then the third carbon, if we look right here, we have an OH that is pointing above the ring. So it's down, down, up. And then finally we have the fourth carbon here, and we have down. So it's down, down, up, down. Okay? So let's take a look at, again, that structure that we just looked at in 2 dimensions. So if you recall, it was down, down. So down, 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 up, down. And then the CH2OH is always pointing up. So this is alpha glucose, what we were looking at. If we go up, down, then that is going to be beta glucose. And they're different isomers. They're going to have different structures and different functions. So one of the things you're going to have to do is know the difference between alpha glucose and beta glucose and know how to draw them. And the way that we remember alpha glucose is down, down, up, down, or dud. Okay, so down, down, up, down, dud. And that will help us draw it. So let me show you what I mean. We want to draw our 6 carbon ring, our 6 carbons and our hexagon ring with the oxygen in it. We start by making this shape. We then put 1 carbon off to the top. So this is 1, 2, 3, 4, 5, 6. And what we do is we want to do alpha glucose. We go dud, down, down, up, down. Starting at number 1 carbon, we put an OH down, we put another OH down. That's the down, down. And then we go up, and then we go down. And now we have our alpha glucose. If we want to do beta, it's just the same thing, except it's a dud. So the OH would go up. And now we have beta glucose, which again is a different molecule. It has a slightly different structure, but that whole difference is going to make a huge difference in biology. That different shape makes a big difference in biology. Now there's other monosaccharides, and you don't have to draw all of them, but you should know that galactose looks almost exactly the same as glucose, but it is down, down, up, up. And that is what is found. Galactose is the sugar that's found in milk. It's completely different. And your body can still metabolize it, break it down for energy, but it's that milk sugar that you find. Now, fructose, found in fruit primarily, right? This one is going to be a 5-ring carbon, and it's slightly different as well. So it is isomer as well. So these guys are isomers, different structures, different functions. Disaccharides form when two monomers come together and form a glycosidic linkage. A glycosidic linkage is the linkage between two carbons, so a carbon on one glucose and a carbon on another glucose, and oxygen's in the middle. So it's really an ether linkage, if you remember back to the organic part of this unit. And these are two glucoses because, again, they're following the DUD. So we have two glucoses that are, alpha glucoses, and they're forming at the 1,4 carbon. And this glycosidic bond, again, you're going to have to know how to draw this, happens when water is removed, and it is an alpha bond when it's 1,4. Our body can digest this. So we can digest it. We're going to find out that we can't digest what we call a beta glycosidic linkage. This is an alpha one. So what is an alpha glycosidic bond? Well, it's when they both point down from the ring. So when you have two glucose coming together, they're both pointing down. Our bodies can digest that. Another example of this is sucrose. Sucrose is table sugar, and an alpha glucose combines with a beta fructose. The OH will both be pointing down, and you'll end up getting this alpha glycosidic linkage. This is an example of a beta glycosidic bond, and here we have one OH pointing up off the plane and one OH pointing down, and this is not when they're both pointing down. So it's not an alpha, it is a beta when one's pointing up and one is pointing down. So this one is pointing up right here, and this one's pointing down. Now that's an alpha glucose directly above us. This is a galactose. We talked about that briefly, and when they come together, they form this type of bond, and our body actually has a tough time digesting it. A certain percentage of the population lack an enzyme called lactase, which is found in most young mammals, but a certain group of people, like depending on your background, you may lack lactase, and that prevents you from breaking down lactose, and you're lactose intolerant. Sometimes instead of showing it like one's going down and one's going up, they show it as a Z. And this Z is showing us that, hey, this is a beta linkage that exists in that disaccharide. When we talk about polysaccharides, it's when we take not just two monosaccharides and combine them, but we form many monosaccharides to form polysaccharides. And the most common polysaccharides that you need to know formed from glucose are starch, which is found in plants, glycogen, which is found in people, and cellulose, which is found in plants. So starch is, if you can take a look, it's an alpha 1,4 glycosidic bond. It's used in plants, and when we eat starch, we can easily break down the starch part of a plant. So if you eat a potato, mostly starch, your body can easily generate that and split that up into little glucose and use that in cellular respiration for energy. Your body will store glycogen. This is a glycogen molecule. We're going to learn that it has many branches to it. So it also has alpha 1,4 glycosidic bonds, and every once in a while it will throw a 1,6 glycosidic linkage, and your body can easily chop this up. As soon as you get a lot of adrenaline, your body will start releasing massive amounts of glucose from glycogen, because the enzymes can easily access all these different points, which are glucoses just available for the enzyme to chop off really quickly. And you get a huge burst of energy when you get your adrenaline being released. Cellulose, on the other hand, has a beta 1,4 linkage. And you can't break this down. So if you, as much as you'd like to eat, get energy from a plant, you can't, because it contains this, this beta 1,4 bond. All right, what I'd like us to do next is look at this worksheet, Carbohydrate Worksheet. Take a look at it over. What you have to do is you have to fill in what molecule is used by plants for energy, what molecule is used by plants for structure, what molecule is used by animals to store glucose, okay? What polysaccharide? What polysaccharide is used for animals, particularly shellfish, to create their structure? Practice these questions, complete this, please. You have to draw glucose. You have to talk about the differences between starch and cellulose. You have to draw an alpha 1,2 linkage. You may need a little bit of help with this, but give it a try. On your carbohydrate linkage worksheet, what you need to do is you need to identify glucose, galactose, or mannose as the monomers making up the disaccharides or polysaccharides. So you have to look up what mannose looks like, how the OHs are facing, up or down, and just basically label those on this diagram. Okay, and that's pretty much it. Oh yeah, one more thing you have to say if it's an alpha 1,4 or a beta. So an alpha linkage or a beta linkage. Pretty straightforward, I hope. And I look forward to seeing you guys tomorrow. Thanks very much.